this is Dr. Bond, and we're going to talk about the electron transport chain and chemiosmosis in this series. It's like how much ATP do we get from this one molecule that we're oxidizing and decarboxylating. I'm going to write it all out for you. My writing is atrocious, and for that I apologize. Kind of. I can't really help it. This would be my cell. And inside the cytosol, here's our organelles. Those are horribly drawn mitochondria. So inside of this cytosol, we have glucose coming into the cell, or possibly, like if this is a liver cell, it was broken down into glucose. But once it gets into the cell, we're going to convert it to glucose 6-phosphate, which happens to be the first step of glycolysis. We're not going to talk about glycolysis right now, but from here, we know in the process of glycolysis, we're going to go from a 6-carbon glucose to 2 3-carbon pyruvate molecules. So this happens in the cytosol. So the glucose comes in, it turns into glucose 6-phosphate, or it gets phosphorylated, and this is the process of glycolysis. This is actually step 1 here. And then we have the 10 steps of glycolysis. We end up with 2 3-carbon molecules of pyruvate. We haven't lost any carbons yet. This has a net ATP of 2. From glycolysis in steps 1 and 3, we had to use 2 ATPs, but we eventually create 4, so we have a 2 net ATP. We also have 2 NADH plus H, and this is the reduced form of the electron carrier known as NAD. These electrons that are carried from NAD will go into the mitochondria to the ETS via a shuttle. There's 2 different kinds of shuttles. We won't talk about them now, but just know that these NADs do not go into the mitochondria, but rather they're going to shuttle their electrons through. Ultimately, these electrons from the original glucose molecule do make their way into the ETS. Here is the ETS. Okay, so these, let me go ahead and go do it like this. Okay, the electron transport system. Okay, so this molecule of pyruvate is going to travel inside of a mitochondria. Okay, it will find its way into a mitochondria, and from there, so in here is our 3-carbon molecule of pyruvate. So this pyruvate is going to have to be oxidized before and decarboxylated before it goes into the Krebs cycle. So we'll talk about that in a minute, but let's just overview. It is going to be combined with a coenzyme A molecule. We're going to lose a CO2, and that is ultimately what you're exhaling, so that is gone. And so we have the 2-carbon acetyl-CoA molecule. This is what enters the Krebs cycle. We can also get an acetyl-CoA from fatty acids, ketone bodies, or cholesterol. So acetyl-CoA we can also get from fatty acids, ketones, and cholesterol, or from carbohydrate metabolism. Here we have an acetyl-CoA. This is going to enter the citric acid cycle. The citric acid cycle has 8 steps, and what its function is to completely oxidize glucose. It will, for each glucose molecule, we have 2 turns of the citric acid cycle. So we do have a NAD produced, a reduced NAD from this oxidative decarboxylation here. Again, we'll go on details in a minute. The citric acid cycle, we're going to have a net produced. We have 6 NADH plus H, 2 FADH2, and 2 CO2 molecules and 2 ATP molecules. From the citric acid cycle, our electron carriers are going to all drop off here at the ETS, the electron transport system. So let's go there. So this is where the ETS will take place, right here. The ETS is going to have 4 complexes, and of these 4 complexes, we will be pumping protons. Okay, so 1, 3, and 4 are proton pumps. And what they're doing is they're setting up a hydrogen ion gradient. From here, there's only going to be a smaller amount, but they're pumping these against their concentration gradient. Okay, so we have a higher amount of hydrogen here, so the pH therefore is going to be lower. So we set up an electrochemical gradient. Okay. Now, NAD is going to drop off the electrons at complex 1, and we'll talk about what these complexes are and how they work. NAD drops off at complex 1. We have a FAD is dropping off here, but FAD is already in this membrane, so we'll talk about that. But FAD is going to drop off at complex 2, which is not a proton pump. Both of them are going to be giving their electrons to coenzyme Q. From here, we go to complex 3. We have another mobile carrier, which is actually going to be better drawn here. And this is cytochrome C. So this is another mobile carrier. And then we go to proton 4, proton pump 4. Then we go to proton pump 4. And from here, we're going to produce water. Okay, we'll discuss the details on that. All right, so what do we do with this gradient? So here, we have ATP synthase, which is an enzyme that's actually a channel protein. And what will happen is these hydrogens will diffuse through this channel back into the matrix. And when they do that, we're going to catalyze that reaction and create ATP. We're dumping ATPs into the matrix. Okay, enough of an overview. Let's get to the actual steps. Okay, first we're going to talk about pyruvate oxidative decarboxylation. So as I just discussed, we had glucose coming into the cell. It becomes glucose 6-phosphate. It undergoes glycolysis if we need to produce energy for the cell. Say our cell's low in energy, which would a high concentration of ADP and a low concentration of the reduced form of NAD would be an indicator. So in any case, we have these pyruvate molecules. If there's oxygen in the cell, the pyruvate is going to go to the mitochondrial matrix. So this happens in the mitochondrial matrix. What exactly happens? We have pyruvate, which is a 3-carbon molecule, and we are going to be reacting with thiamine pyrophosphate. We're going to be decarboxylating, so we're going to lose a carbon, and we're going to end up with hydroxyethyl TPP, or you can also say HETPP. And then this is, of course, the cycle. This happens in the first enzyme of the pyruvate dehydrogenase complex. Pyruvate dehydrogenase complex, E1 is pyruvate component. So this is pyruvate dehydrogenase component, which is the first enzyme of a series of 5 enzymes that are in the pyruvate dehydrogenase complex. Okay, so this is, we'll say, step 1, enzyme 1. Okay, again we have pyruvate reacting with thymine pyrophosphate. We're creating a molecule of hydroxyethyl TPP, which is here. And so this is the product of step 1, HETPP, and we lose a molecule of CO2. Okay, so that's step 1. This happens in the mitochondrial matrix, and this is not part of the Krebs cycle. This is a pre-step before we enter the Krebs cycle. We're going to be in enzyme 2. Enzyme 2 is dihydrolipoyl transacetylase. So what we were left with, this was the product of step 1. So here, we're going to be reacting with, let's see, lipoic acid. What's important here is this disulfide bridge. This is going to be the oxidized form, and we'll say it's relaxing. It likes to be it likes to be in this form. That's the relaxed form. But we're going to be reacting it with the HETPP. Okay, when we do, what we're going to end up with is so this is going to be our acetyl group that will attach to the lipoic acid, one of the sulfurs. We're going to break this disulfide bond, and we reduce the other sulfur. The next step of this, so it would be step 3, coenzyme A, which we draw with a sulfhydryl group, because this is going to be our reactive group here, functional group. It is going to take this acetyl group away. So we're going to make an acetyl-CoA. That will then proceed to the citric acid cycle. When that happens, this is the form. This is, once we lose the acetyl group to the coenzyme A, and we have the acetyl-CoA and it leaves to the citric acid cycle, this is the form of lipoic acid that we're left with, which is a reduced form. We call it dihydrolipoamide. This can't react anymore, and this is an enzyme, so what we're going to have to do is oxidize that in the next step. So for a review, enzyme number 2, dihydrolipoyl transacetylase, just think about what it's doing. It's transferring an acetyl group. Not only does it transfer it once, but twice. The first time we're going to take the acetyl group, we're going to take this part, and we're going to give it to lipoic acid. When we do, it's a 2-step process. We're going to reduce one of the sulfurs. The other sulfur is bound to the acetyl group. From here, coenzyme A comes in, and takes the acetyl group and makes acetyl-CoA, and it leaves to the citric acid cycle. Here we have the reduced form of the lipoic acid, which is dihydrolipoamide. This is going to be involved in the next enzyme in the next step. So the actual product of this reaction would be acetyl-CoA, and then our coenzyme, which is now reduced. Our coenzyme here was lipoic acid. The lipoic acid is basically going to act like a swinging arm in between enzyme 1 and enzyme 3. Dihydrolipoyl dehydrogenase is the enzyme number 3, or you can say E3. And from here, what we want to do is get rid of these hydrogens. So they're going to first give it to FAD, but FAD is not a mobile electron carrier, but NAD is. So NAD comes in, takes them from FAD, and then this guy. Okay. So what we've done is oxidize the lipoic acid. Okay? So our products in this case would be an oxidized form of lipoic acid and a reduced form of our electron carrier, NAD. Okay, so this is the pyruvate dehydrogenase complex, and the products overall, we got CO2, one molecule of CO2 from the first step. We have acetyl-CoA, and we have a reduced electron carrier, NADH plus H. Okay, so this is our products of pyruvate oxidative decarboxylation, which happens in the mitochondrial matrix. This goes to the citric acid cycle. This will go to the ETS. This ultimately is a byproduct, and we're going to exhale it. It does go through the blood, the bicarbonate buffer system, or it can just be dissolved in blood, but ultimately you're going to exhale. So this is a byproduct. We don't do anything else with that. Plants can fix it. We don't do anything else. Here we have acetyl-CoA and then the reduced form of our electron carrier. Okay, so we had 5 components of the pyruvate dehydrogenase complex. We have E1, E2, E3. These are catalytic, the E1, E2, E3. The first one, pyruvate dehydrogenase. We can say component. Second one, dihydrolipoyl transacetylase. Then we have a kinase. The kinase is going to be undergoing phosphorylation. The phosphatase undergoes dephosphorylation, and these are regulatory. So these are going to be controlling these enzymes, particularly this guy. This is a complex that is going to be very similar to the citric acid cycle, alpha. Okay, so alpha ketoglutarate complex of a CAC, step 4, very, very, very similar. Okay, the coenzymes of this complex, remember in pyruvate dehydrogenase, the coenzyme was TPP. In dihydrolipoyl transacetylase, the coenzyme was lipoic acid. Then the coenzyme is a vitamin-derived FAD. And then, of course, we had NAD working, and we had coenzyme A working along with it, too. So we have NAD and coenzyme A that also are part of this, or work with this complex. a summary of what happens in the pyruvate dehydrogenase complex. It happens in the mitochondrial matrix. Pyruvate loses a CO2. Hydroxyethyl TPP is formed. A hydroxyethyl to lipoic acid oxidized to acetyl will be oxidized to acetyl dihydrolipoamide. Coenzyme A comes and steals the acetyl group and goes off to the CAC. And then we are going to reoxidize dihydrolipoamide to lipoic acid, thereby reducing NAD, which goes to the ETS.